All right, so great. Yeah, thank you very much, everybody, for joining and taking time to listen to my talk. My name is Sungsik Kong, and please feel free to call me Kevin. I'm currently a postdoctoral research associate at Wisconsin Institute for Discovery, affiliated with the University of Wisconsin-Madison. Today, I'm going to talk about one of my main research interests, which is how we can estimate phylogenetic networks from genomic data using composite likelihood. So this is a phylogenetic tree, and it has been useful for almost any biological investigations. Here I show a very simple example of a phylogenetic tree that contains 5 species, where each of the species are called leaves labeled on the bottom. And the relationship of these 5 species begins from the root, which is a single point on top that represents the most recent common ancestor of the species under consideration. And in between the root and the tips, we have a series of internal tree nodes where a single branch splits into two. It denotes an ancestor gave rise to all of its descendants below that internal tree node. And it also represents a speciation event where a single ancestral species splits into two descendant species. This phylogenetic tree, again, has been useful in many fields, for example, epidemiology, classification, and systematics of biodiversity. But the question is the phylogenetic tree a perfect model? Unfortunately, no, because phylogenetic trees often overlook some of the important biological processes that also can result in the production of a novel species, like hybridization, that refers to interbreeding between two distinct species. And this hybridization has been historically neglected. It has been seen as something unnatural, a rare biological event. And even if it somehow happens in practice, its offspring was not expected to persist over evolutionary time. So there was a rationale that we can simply ignore this presence of hybridization in biological studies. But thanks to the recent accumulation of the genomic data and development of computational methods to detect hybridization, we now know that hybridization is much more common than we previously thought. And to support this argument with some numbers, some papers argue that 25% of plants and 10% of animal species probably have experienced hybridization at some degree during their evolutionary history. One way to conduct a phylogenetic analysis in spite of this hybridization is we simply ignore their presence and stick with the tree analysis. But unfortunately, such practice will result in a biased species tree estimation, which means no matter how much time and resources you spend for the reconstruction of the species tree, or how much data, how much data you have for that reconstruction, the resulting species tree will be wrong. So it's going to tell us the incorrect relationship and history of among the species in interest. And to overcome this limitation, a new concept called phylogenetic network has been proposed. And phylogenetic networks and trees are similar in a way that they both are composed of set of nodes and the branches that connects those nodes together. But the networks are distinguished from the trees by this reticulation structure, where two branches that are once separated to merge back at a single node called the reticulation node. And here I marked in a pink circle here. And this acyclic structure denotes a hybridization event. And so in this cartoon example, I show this reticulation shows that species 4 is the hybrid daughter of species 3 and species 5. But in practice, two types of networks are being used. And one is abstract network, which an example is shown here on the left side. And another one is explicit network on on the right side. And two networks look very different from each other, and that's how much difference there are in terms of their algorithm to compute one and the biological interpretations that you can make based on these networks. To be more specific, abstract networks usually result in more colorful diagrams because their tips or their leaves are shaped in circle and they're colored using some sort of external information, for example, geographic location, where the sequences were sampled. So based on those colors, we can understand the geographic locations, the pattern of geographic locations of occurrence among the sequences. And the abstract networks are usually very large in size, and this is because they are fast to compute. The reason is most of the methods use distance-based optimality criteria. So it's very fast to compute an abstract network using very large data set. And abstract networks are typically unrooted. It lacks a rooting process for its computation, which means we can't understand the ancestor-descendant relationships between the sequences or between the nodes, which is probably the most basic information that you want to extract from any phylogenetic analysis. So the goal of this abstract network, the point of this method, is to display the pattern of the nucleotide arrangement among the sequences and to display the conflicting signals in the data in the form of cycles. On the other hand, explicit networks on the right side resembles very similar to a phylogenetic tree. And again, the only difference is it has a number of horizontal branches that denotes complex evolutionary histories. And they are small in size because many of the methods that infers explicit networks are model-based, which means it is computationally very intensive to reconstruct one. So compared to abstract network or the phylogenetic trees, explicit network usually result in very small size tips or very small size network. And explicit networks are often rooted, which means we can understand the timing of the speciation event and timing of the hybridization event based on the network. But in some cases, some explicit networks are semi-directed, which means there is no directionality among the tree branches, but there is a directionality among the reticulation branches. So at least we can know who are the parents and who are the daughters in the hybridization event. So the goal of these explicit networks is to infer complex evolutionary histories based on some model. So I hope I made it clear that abstract networks and explicit networks are two different things. And explicit networks are the ones that we want to use in biological investigations, and abstract networks are the ones that we want to visualize the patterns in the data. But in practice, abstract networks are very popularly used to make some biological inferences. And here I highlight one of the examples that happened in the middle of pandemic. So in the beginning of pandemic, a paper was published in PNAS with a very ambitious title saying Phylogenetic Network Analysis of COVID-19 Genomes. And in that paper, they had a single figure, which is a median joining network, which is one of very popularly used abstract network, which is shown here. It was great to see some people are working to understand the evolutionary history of the virus in the beginning of pandemic when we virtually didn't know anything about the virus. But the problem was the authors tried to overinterpret this structure, and they made interpretations to understand the origin of the COVID-19 and COVID-19 pandemic, as well as to trace the transmission history. And this was rather frustrating because such misinterpretation or overinterpretation will only make us having difficulty to effectively combat the virus. So myself, along with some of my colleagues, we submitted a rebuttal letter with a very strong title to make things as clear as possible. And we said a median joining network analysis of COVID-19 genomes is neither phylogenetic nor evolutionary. And we try to make 2 points. First, because median joining network is an unrooted structure, we can't understand ancestor-descendant relationships between the viruses. Therefore, we cannot understand the origin of the pandemic. Secondly, even if it somehow tells us a true relationship between the viruses, relying on a single phylogenetic tree or a network is not the best practice to understand the transmission history. So what should have they done? They should have inferred on explicit networks. And in the past 20 years or so, mathematicians and statisticians, biologists, and computer scientists has been working hard to understand the structural properties of these explicit networks. And we now have more than 20 subclasses of these explicit networks. And each of these subclasses and their biological significance has been reviewed very nicely in this review paper that is shown here on the top right corner. In this paper, we had a single figure, which a single diagram, which is shown here, where each of the nodes represent a distinct subclass of explicit networks, and these subclasses are linked by an edge based on their structure similarity. However, many of these subclasses are originally proposed to understand the mathematical characteristics of the network rather than to be directly applicable to the biological investigations. For example, these k-nested networks, it shows very complex graphical representation to connect different nodes together using reticulation, but it's computationally difficult to infer this kind of networks. But among these subclasses, I'd like to highlight two of the subclasses that has been very useful in biological investigations. And one is tree. This is the phylogenetic tree that we typically use, which is simply a special case of explicit network where the structural property is lack of reticulation. But more interestingly for us network people, we are interested in using these level 1 networks. Level 1 networks are very similar to the trees in terms of structure. It has reticulation, however, but we assume the reticulations are occurring independent from each other, or the reticulation does not share a common edge, which assumes biologically that hybridization events will occur independent from each other. And the biggest advantage of using these level 1 networks is for computational tractability. And many of the computational methods that tries to estimate phylogenetic networks from genomic data assumes that the true network or true relationship between the species can be represented in level 1 networks. And today we have a handful of computational methods that estimates phylogenetic networks, and they can largely classify it into summary-based methods, which take a two-step process. First, they summarize the raw sequence data into a set of gene trees and use those set of gene trees as input to estimate the phylogenetic network. And these methods, the biggest advantage of these methods is that they're relatively fast because a lot of hard work has been done prior to the network estimation. But the downside is that their accuracy completely depends on the quality of those estimated gene trees rather than the quality of the raw sequence data or the algorithm itself. And we have a number of functions that does this implemented in Phylonet that directly uses rooted gene trees, or SNAC, which extracts what's called quartet concordance factors from the set of gene trees and estimate a phylogenetic network. Another class of computational methods to estimate phylogenetic network is full data method, which directly takes the sequence data as input instead of inferring the set of gene trees from the sequence alignment in prior to the analysis. The advantage of this method is that they utilize all of the signals that exist in the alignment, but the downside is that they are computationally very heavy, limiting their use to a very small data set and for very small scale biological investigations. And many of the methods uses Bayesian framework, which tries to simultaneously estimate the gene trees and the species network. But today we're going to talk about this new method called FINIST, which I proposed recently, which tries to overcome the limitations in both classes of network inference methods. And this FINIST, by the way, is the acronym for phylogenetic network estimation using site patterns. So the main question for FINIST was that can we estimate a phylogenetic networks directly from the sequence data, bypassing the gene tree estimation step as in the summary-based method, efficient as the summary-based method, and as accurate as the full data methods? And long answer short, fortunately, my answer is yes, we can. And we do so by combining what's called site patterns and the composite likelihood framework. So what do I mean by site patterns? Site pattern is the assignment of the nucleotide states at the tips of a topology. So let's consider this cartoon network example on the left side and the corresponding sequence alignment on the right side. If we select the very first column of the sequence alignment and we map that onto a topology to the corresponding species, we get the site pattern of AAAAA that tells us that there is no variation between the 5 species at that specific site. We can do the same thing, for example, for the 5th column of that alignment, and now we will get the site pattern of TTAAT that tells us a bit more story than the previous case, because species 3 and 4 shares the same nucleotides that is different from everybody else at the specific site. At this point, you might wonder, okay, this sounds very similar to the SNP data. Are those same thing? Actually, each of the sites in the site patterns are called coalescent independent sites, and it assumes that every site has its own distinct evolutionary history. And the biggest difference between the site patterns and the SNP is the site pattern also utilizes those sites, the information from those sites, where there was no variation like we see in site 1 in this example here. So the site patterns and the SNPs, they might sound similar, but they are two different things. So for the next few slides, I'm going to talk about how we can use these site patterns and how we can compute the composite likelihood of a phylogenetic network using this simple example here. Here again, we have a network that has 5 species and a single reticulation where species 4 is the hybrid daughter of species 3 and species 5. But before I go further, I'd like to formally introduce a number of parameters that we are interested in estimating in our method. And first parameter is called tau, which represents a set of speciation times that are measured in coalescent unit backward in time. And each of this element in tau is assigned to each internal tree node. And because it's measured in backward in time, its value equals to 0 at tips, and the value increases as it moves towards the root. And each element in tau has some biological meaning. It represents the timing of the speciation event because we only assign them to the internal tree nodes. The second parameter is gamma, which is also often called inheritance probability or hybridization parameter, that is assigned to one of 2 incoming branches towards the reticulation node. And because gamma is a probability, its value ranges between 0 and 1, which in turn makes the probability of the other incoming edge as 1 minus gamma. And gamma also has a biological meaning. It tells us the proportion of genomic contribution from one of the parents towards the hybrid daughter. So this blue gamma we see in this network example here tells us the proportion of genomic contribution from species 5 to the hybrid daughter species 4. And finally, we have theta that denotes a population size parameter, which we assume that there is a single population size throughout the entire topology, which means there is a single value for theta in every topology. So first thing that we want to do is we want to break the network down into set of displayed trees. And we can get displayed trees by removing one of 2 reticulation branches at a time. And each of the display tree will have the probability that can be computed by multiplying all of the inheritance probability that are assigned to the reticulation branch, which are not deleted in the production of that particular display tree. So here our network N can be decomposed into 2 display trees, where display tree 1 has a probability of gamma, and display tree 2 has the probability of 1 minus gamma. And for each of the display tree, we further decompose it into a set of quartets, which refers simply to a tree that has 4 tips. And we do so by selecting 4 tips from our display tree at a time, along with some of its internal tree nodes that represents the most recent common ancestor for the pairs of taxa, and the branches that connects those nodes together. So for example, here, by selecting species 1, 2, 3, and 4, along with some of its internal tree nodes and the branches, we get our first quartet here, and we can repeat this process for every combinations of 4 taxa to extract 5 quartets from our display tree 1. And of course, we can do the same thing for the other display tree to extract another 5 quartets from our second display tree. And each of these quartets will also have the probability that is assigned to the display tree that the quartet was decomposed from. Now, zooming into one of the quartets, since at a given tip and at a given site, each tip can have any one of 4 nucleotide states, which means there are 4 to the 4 or 256 site patterns can arise on any quartet tree. But the good news is the probability of any one of these 256 site patterns that arise on a quartet can be computed under a simple model called Jukes-Cannon model. And what's even better is these 256 site patterns can be generalized into 15 patterns. So letting I, J, K, and L denote distinct nucleotide states, A, C, T, and G, the 256 site patterns we see in this matrix above can be generalized into 15 patterns we see in the second row here. And we're going to call this vector as PQ. And this true site pattern probabilities can be computed using the formula that is presented in Chiefman and Kobatko 2015 for any quartet given the set of speciation times tau and the population size parameter theta. And now, given that we can compute the site pattern probabilities for any quartet tree, we can extend this idea to compute the true site patterns for any quartet that is extracted from the network. So let's say we extract a quartet that has a reticulation, which we call Q, shown here on the left side. The way to compute the true site pattern probability for this quartet network is simply the weighted sum of the site pattern probabilities that are computed for the quartet that has same 4 tips, each of which arises in each of the display trees. And when I say weighted sum, by the weight, I mean the probability that is assigned to each of the quartet that is decomposed from each of the displayed tree. And now we have a way to compute the true site pattern for any quartet network. The next thing we want to do to compute the likelihood of this quartet is to get the observed site pattern frequencies from the data. And we do so by selecting a subset of sequences from our original data set, and by subset selecting 4 sequences that also arises in our quartet of interest. So here on the right side, we selected species 1, 3, 4, and 5 because they also occurs in our quartet of interest. And starting from site 1, we identify which one of 256 site patterns it belongs to. Then we identify that site pattern to know which of the 15 generalized site patterns it belongs to, and we count the number of occurrences from the beginning to the end of the alignment. So at the end of this process, we will get a vector that has length 15 and where each element is an integer, and some of these elements will equal to the sequence, the alignment length of our data. So given that we can compute the true site pattern probabilities from the network topology, and we're going to call that PQ, and we can get the observed site pattern frequencies from the subset of data, which is called WQ, we can get the likelihood of a quartet. And by multiplying all of these quartet likelihoods together, we can get the composite likelihood of a network. And at this point, you might say, but is this actually the right way to get the likelihood of a network? Because the quartet that is extracted from a common network is not independent from each other. That is actually right. The quartets are not independent from each other, and this is why this is called composite likelihood, or is also called pseudo likelihood. But approximating a likelihood of a network topology or tree topology using this approach has been shown to be performing quite well in both phylogenetic tree and network estimation conditions. So we can stick with a composite likelihood of a network to approximate the likelihood of a network and save lots of computational time instead of trying to compute the full likelihood of a network. So now we have a way to evaluate the fit of the data onto a topology. Our next step is to find the topology that describes the data the best. And to do that, we need to do heuristics. So to explain how the network space works, let's consider this simple cartoon example of the network space that has two peaks. And we're going to call the lower peak as local optimum, and we're going to call the upper peak as global optimum. And any point in this network space is a topology, and higher the altitude of the topology, higher the likelihood. So the idea is we start at a certain point in this network space, somehow we traverse around, and our goal is to eventually get to the global optimum in this network space. But of course, this is a very simplified representation of what the network space actually looks like, because every time we add a reticulation, or every time we increase this parameter h, the dimensionality increases, which means there is a whole new space of networks that needs to be searched, that has increased number of reticulation. And what's more complicating is every time we move up the reticulation, the topology at the global optimum in that upper dimension of the network space also increases. So unless we limit the number of reticulations to be included in our final network, there will be two problems. First, the search will go forever. The search will never stop because it's going to continuously find the topology that will describe the data better by adding more reticulation. And even if it somehow ends its search, the final network will have excessively large number of reticulations, which will be very difficult to make any biological interpretations. So to prevent this problem, we need to pre-specify how many reticulations do we want in our final network. But unfortunately, there is no straightforward way to identify this value, the number of expected reticulations in the final network. And this is where the information about the biological system comes into very important play. Anyway, to efficiently search the network space, we have implemented two searching strategies. And first one is called hill climbing. So the idea is we start at a certain point in this network space, and we're going to call that starting topology. And this starting topology can be either randomly generated, or it can be estimated using some tree estimation method. We're going to compute the composite likelihood of the starting topology, and we're going to make a little tweak on the topology and propose a new topology. If the composite likelihood of the new topology turns out to be higher than the starting topology, we make an upward step in the network space. And if the proposed topology turns out to have worse composite likelihood than the starting topology, we discard the proposer and we make another tweak onto the starting topology to make another proposed topology. We continuously do this process until we get to the global optimum, or until we get to the sum peak where we cannot make any upward movement anymore. But because hill climbing only makes upward movement, upwards traversal of the network space, depending on where you start your network search, there is a high risk of becoming stuck at the local optimum, which is another peak in the network space that is not the global optimum. And we want to avoid this problem because we are not going to find the best topology that describes the data. To prevent this problem and to eventually increase the probability of finding the global optimum and decrease the chance of becoming stuck at the local optimum, we have implemented our second strategy called simulated annealing. And simulated annealing searches the network space in a way by allowing the downward movement at some probability. But the downside is because simulated annealing searches the network space much more exhaustively than the hill climbing algorithm, it's going to make many more number of steps to search the network space, which can be directly translated to increased computational time. But it has higher chance of finding the global optimum. So all of this method has been implemented a Julia package called Finest, and we tested their performance using simulated data. And for simulation, we considered this species network shown here that has 7 species and 2 reticulations, where species 2, 3, and 4 forms a reticulation, and 5, 6, 7 and forms a reticulation. Under this species network, we simulated a set of gene trees that ranges from 100 to 5,000. And for each of these gene trees, we simulated sequence alignment using a little more complex model than JC. We used HKY model. And using those simulated sequence alignment, we estimated a set of gene trees. So at this point, using the simulated sequence alignment, we estimated the network topology using finest using hill climbing algorithm, and finest using simulated annealing algorithm. And using those estimated set of gene trees, we estimated network using Phylogenetic using maximum pseudo likelihood framework and SNAG. And we evaluated their accuracy by comparing the estimated network with the true topology that is shown here. So this plot summarizes the topological accuracy of the 4 methods, where each of the methods are colored using different color schemes. The x-axis represents the data set size in terms of number of gene trees, and the y-axis represents the dissimilarity. Higher the value, more different that the estimated network from the network that the data was simulated on. So when the number of gene tree was small, all 4 methods did not perform very well. And among those, Phylogenetic performed the worst, whereas both finest using hill climbing and simulated annealing performed quite better than the other 2 summary-based methods. When the number of gene trees were 500 or 1,000, all 4 methods performed quite similar to each other, but finest using simulated annealing performed better than the rest of them. And this superior in accuracy in finest using simulated annealing was more highlighted when we had more number of gene trees. When the number of gene trees were 3,000 and 5,000, Phylogenetic using maximum pseudo likelihood framework continues to performing the worst. And SNAG and finest using hill climbing algorithm performed quite similar, and finest using simulated annealing performed the best. And how about the computational cost? This plot summarizes the computational cost of the 4 methods. And for now, let's ignore this red bar here. The x-axis again represents the dataset size in terms of number of gene trees. Y-axis represents the time taken to estimate the network in terms of hours. We can see many of the methods except blue that represents finest using simulated annealing algorithm clustered close to x-axis, which means all 4 methods are very efficient to compute the network, including finest using hill climbing algorithm. But focusing on this blue plot here, which takes about 20 to 30 hours in average, we might think, okay, finest using simulated annealing algorithm, it might sound like it turns out to be giving the best network or most accurate network in cost of computational times. Well, that might be true. But the computational time to estimate a network using simulated annealing actually is more efficient than computational time that is required using summary-based method if we consider the time that is required to estimate the set of gene trees to even initiate the summary-based method. So these red bars represent the time that is required to summarize the sequence alignment into set of gene trees. Until the point when the number of gene trees were about 3,000, it is true that the finest using simulated annealing take more time than any of the summary-based methods, even if we include the time that is required for the summarization step. But when we have the number of loci 5,000 or more, the time that is required to summarize the alignment into set of gene trees exceeds the time that is required to estimate the network using simulated annealing algorithm. So we can say when the dataset size is small, finest using simulated annealing algorithm might be the best way to get the most accurate network using the least computational cost. You might say, okay, but this estimating the set of gene trees can be parallelized, and this computational time to this summarization step can be improved. But it is true only when the resources are available. So, and we also tried finest on a number of empirical data sets. And here I show an example using Papio-Nini primates. Here we see 7 species and a multiple hybridization event or gene flow events were observed between the species of this Papio-Nini group. And the integration or gene flow among these species were detected using what's called gamma, which is similar to the ABABABA test, but using gene trees. Using this genomic data set, we estimated 2 phylogenetic networks, assuming there is 1 hybridization event or 2 hybridization events. When we assume that there is a single hybridization event among these 7 species, we observed a single reticulation event among the African species. And when we increase this number of expected reticulation to 2, this reticulation among the African species was retained, and we additionally observed another reticulation event among the macaca species that occurs in Asia, which aligns with the biological knowledge that we know, because we know macaca species experience lots of gene flow in their history. So in summary, phylogenetic, I hope I convinced that phylogenetic networks better represent non-tree-like evolution scenarios than trees, and we must start to use phylogenetic networks more frequently than the phylogenetic trees. And more importantly, if we want to conduct a network analysis, we should use explicit networks rather than the abstract networks. And in this talk, I show how finest phylogenetic network estimation using site pattern works, which is a novel method, novel network inference methods that uses site patterns to estimate the network. And this is publicly available on GitHub repository here. And currently, it takes phylip sequence alignment, and it yields phylogenetic networks in terms using in the extended Newick format. And the biggest advantage of our method finest is that it bypasses data summarization step, and it utilizes all of the phylogenetic signals in the alignment. So what's the next? Finest still has a number of gaps that needs to be filled to be applicable for the wide range of biological systems, even though it can work in many of the cases. The first thing that needs to be improved is finding the root position of the network using site patterns. So currently, because finest estimates root phylogenetic network, it requires user to provide an outgroup sequence. And this outgroup sequence is not always readily available, particularly when you're working with non-model organisms or very large genomic data set. We have a way to find the root position using site patterns for the phylogenetic trees through multiple hypothesis tests, and I'm planning to extend this idea to find the root position of the network using site patterns. And secondly, there are a number of assumptions that might be biologically unrealistic to improve the computational tractability and computational speed. And these assumptions include the constant effective population size and assuming that there is a single individual sample per species. And relaxing these assumptions will require some of the proofs and some of the improvements in coding the software. But hopefully this can be fixed in the short future. And finally, because we are assuming the true relationship between the species can be represented on the level 1 networks, we tend to oversimplify what actually happened in the history. So our next step is expanding the network space to search networks that are beyond level 1 networks to accommodate more complex biological histories to be represented in the phylogenetic networks. And with that, I'd like to thank a number of my co-authors and a number of institutions that supported my study, including the Ohio State and University of Wisconsin and NSF, and the Academic Society, SSB and SSE, and Kobatko and Solis Lam's lab that supported my research. Thank you very much.
Thank you very much, Kevin. This was great. If anyone has questions, please feel free to, if you want to speak directly, you can raise your hand and we can then unmute and you can ask a question directly. Otherwise, please type your questions into the chat or the Q&A tab. Go ahead, Barty. You should be able to unmute.
Can you hear me? Sorry.
Oh, yes.
Yeah, I just had to go through a series of accepting things. Okay. Um, did you already mention, and if you did, I apologize, but did you mention anything about where does sequence coverage play a role in any of this analysis, if it does?
Yes, for our method, basically we assume that we have an ideal data. So we don't really know if there is a specific sequence coverage to for the methods to perform ideally. But what's more important is how many base pairs do we have? So basically, we're assuming every site is independent observation. So the accuracy is expected to improve if we have longer sequence alignment. And by longer, I mean it's better. It's recommended to have more than 300,000 base pairs, and that's probably my minimum to-go sequence length. But of course, more the better. And sometimes if you try to estimate a network using much shorter sequence, it is very unlikely to find the true relationship.
Yeah. Okay. Thank you.
And then there is a thank you, Bart. And then there is a question in the chat from James Starrett. How does FileNest handle missing taxa in different loci?
Right. So I guess if when you say there is a missing taxa in different loci, if we concatenate all of the loci together, there will be gaps. And I guess at this point, we are assuming that the gaps represents a distinct nucleotide size. So we don't really accommodate the missing data or even the ambiguous data. But there is a way to accommodate that, and we do plan to implement that and study to see how we can deal with the missing data or the ambiguous site in the future. So at this point, we're assuming that there is assuming that every gap represents some meaning, or the fifth text or the fifth state.
There is another question from Ben Romero. Can you explain what means the number above the reticulation branches? What is the difference between if you have 0.6 or 0.2 in those two different reticulation events?
Oh, I see. Right. So the number above the reticulation branch represents the gamma parameter, and some of these two numbers towards a common reticulation node should be some of these two numbers should be equal to 1. And these values represents the proportion of genomic contribution from one of the parents towards the hybrid. So when there is number between, let's say, species, when there is a reticulation branch between species 1 and species, and there is zero point 0.5 on top of that reticulation branch, that means about half of the genome in that hybrid daughter is contributed from species 1. So those are what it means. So when you say zero point 0.6, about 60% of genome was contributed from one of the parents towards a hybrid.
And then there's another question from Catherine Drotos
Yeah.
related to James' question. How does it handle ghost taxa? Taxa that are missing entirely, whether we know about them or not.
Right. Yeah. So to handle that, in the beginning of developing finance, we assumed what's called time consistent network. So which means we assume that all of the players that involve in the hybridization are sampled, and two of the parental nodes that contributes to the hybridization event should have like equal speciation time. But we relax this restriction, and we now allow two parental nodes that are contributing to the hybrid node can be different from each other. And this is very useful when we have ghost data, because when we have ghost data, when we have ghost lineage or ghost taxa, we are missing one of the parents entirely, or we are partially missing them. So we cannot expect two parental nodes to have equal speciation time as the hybrid node. So if there is ghost taxa, we're expecting the speciation times for two internal tree nodes that contribute to the common reticulation node will be very different from each other. And this might imply that there is the presence of ghost taxa, but it can also imply that it represents an incomplete taxon sampling, or the quality of the data is not very ideal.
Manasi is asking, how does the number of taxa analyzed factor in on the computational time taken by Finest?
Right. So for Finest, the nice thing about Finest is that because it uses composite likelihood framework, the sequence length will not increase the computational time. But when we have more number of sequences or more number of taxa, it's going to increase the computational time greatly. And right now, we have tested Finest to infer a network that is about 12 species, 12 taxa. And unfortunately, it did not finish the analysis. It took more than a week to finish a single run. So right now, we are recommending to use Finest if you have 10 or less taxa. And this is not special thing about Finest. Many of the existing phylogenetic network methods that are model-based, the maximum number of taxa it can handle is about 20 to 25. And hopefully every one of them or most of them are trying to improve this scalability, so you can analyze the data set that has many number of taxa. So right now, computational time increases insanely if you have many number of taxa. So its scale is very different from either abstract networks or the phylogenetic tree analysis.
One more question for you from Hannah Kim. Phylonest seems to take a species tree, and that is what it makes, and that is what it's based the solution on for understanding hybridization events. But can it work with gene trees? Would it be looking for recombination events in that case? How divergent the tree should be for both to be done?
Right. Right. Finest uses species tree, and because the biological process that Finest is modeling is a hybridization event, not recombination event. So uh, if you use a gene tree as a starting topology and you use a sequence alignment for a single loci as input, it's probably going to find some network. Is if you set the number of reticulations in the final network, is going to eventually find some sort of a network, but that does not mean the reticulation that we observe in that network represents a recombination event. I think there are a set of methods that actually can infer recombination, probably ancestral recombination graph and so on. So those are more suitable methods if you are trying to understand if you're trying to infer reticulation within the single gene. But for Finest, we are assuming that there is a genome data, and the biological process we are trying to model is the hybridization event. So unfortunately, the gene trees would not work.
But I'm guessing you could use the gene tree as a starting point to.
Oh yeah, oh yes. Yeah, that's right. Yeah, I mean, like for the starting topology, you can even use randomly generated, like completely a star tree. That is also possible. The downside would be that it's going to take much longer time to eventually, find the optimum. So to prevent that, it's better to find something that's more reasonable than the random tree.
And then Barty, you can unmute again, I think.
Yeah. So going back to your previous statement about the maximum taxa to analyze, you said something like 20 or 25. So then what do you recommend if you're trying to have resolve a genus or maybe even subgenus that might contain well beyond that number?
Right.
Which is usually the
Yes.
case. Yeah.
Yeah, that's very that's one of the biggest hurdle, that's something that we want to do. And currently, like what we can do with existing method is, first, usually you estimate a species tree using the sequence or gene tree data. And when you infer a species tree, you will probably conduct a bootstrap analysis, and at some node, that bootstrap value will be pretty low. And, and there are many explanations. Like, there are many things that can explain that low bootstrap support, but one explanation might be hybridization. So if you believe that, okay, probably hybridization is contributing to this low bootstrap support, you might want to extract those species specifically, and then make a subset, make a smaller data set that contains subset of species, and conduct a network analysis. And the rationale to do that, it can be a bootstrap support, but another better way to do it is conduct some other hybrid detection methods like ABABABA test or HIDE, and then you gain more evidence as well as the biological information about the system, and you gain more evidence that, okay, there might be a hybridization among these species here, and you conduct a network analysis using the truncated data set. That's one way to do it. And currently, to bypass that step, because that can be a lot of work, one way is we are trying to work on the improved scalability. So at least for some of the summary based methods that are reliable, I know that they are working on to accommodate 50 taxa, hopefully. And we don't know if it'll actually work or not, but that's like 50 is the goal number that we are trying to do at this point. And another possibility is there is another group of people trying to work on network merging. So what it does is, basically, you have a large data set, you separate these species into different subsets of data, you estimate a network for each of them, and you merge them together to eventually get a large network. So there are different approaches to tackle the problem of not being able to accommodate a large number of species, but hopefully in the coming years that can be improved.
Thank you.
Thank you, Bart. I kind of had a follow-up question on this, because you mentioned that you can use the bootstrap support on the branches, but you in the beginning, Kevin, you mentioned that abstract networks are not super useful for analyzing evolution. And I'm curious if you think in that context they could be useful. They will kind of indicate where on the tree there are those ambiguous areas, and then you can kind of narrow in and do a proper, statistical analysis, and then try to find hybridization.
Right. Yeah, that is true. I mean, yeah, I hope I didn't make it sound too bad about the abstract networks. I do believe abstract networks are very useful if you are trying to understand the patterns in the data, and you try to, visualize some of the different signals in the data. Then you can do that very quickly using abstract methods. The problem is there are a lot of people trying to interpret this split event in the abstract network as speciation event. So they are trying to make inferences that are to be applicable for practice like conservation or epidemiology, simply based on that abstract networks. That's what really scares me. I'm not saying abstract networks are something that we should never use. I'm sure we can make a good use of them if we don't overinterpret them.
Well, thank you very much. I think
I actually had one.
Yes.
Sorry. So have you been able to, as we're talking about this, have you been able to use Finest to see if it better resolves those low bootstrap values generated from a separate tree, to see how it might? So if you use Finest to find these hybridization events, you use a different tool to construct the tree. You notice that there's low bootstrap values at certain nodes, as you had mentioned previously, and then you say, hey, maybe that's a hybridization event going on, and then you try Finest. Have you been able to do a comparison in that way to see if it?
Yeah, not yet. So unfortunately, that's gonna involve we can probably do simulation on that, but if we can use empirical data set, it's going to be more useful. But we haven't tried that, but definitely that might be something. And my guess would be it's very likely that that's what's going to happen, because the reason that there is a low bootstrap support is that every bootstrap replicates, you are getting something different, and this different might be one this difference in the data set can fit better if you have a reticulation event in that area with low bootstrap support. So my guess would be, hopefully, the reticulation will happen in that area where there's low bootstrap. But I haven't tried it myself, so I cannot be 100% sure.
Interesting to see. Yeah.
Right.
Thank you.
Thank you.
Kind of towards that, wanted to ask different, but towards bootstrap support, do you envision, or maybe you already have, adding support on those reticulation events, somehow evaluating it?
You mean like evaluating like support for the reticulation structure?
For every reticulation event, yes, and what's the confidence associated?
Right. So that will be great if we can improve computational time, because right now it's already taking a lot of time to get a network. But for Bootstrap, you need to repeat that many times, right? So if we can improve the computational time, then maybe we can do Bootstrap. But right now it's a little premature to jump into Bootstrap support. But I think some of the summary based methods like Snack, they can find some support values um, for the reticulation at least.
Since you are treating, and sorry that it's stretching a bit long, but I'm just very curious, since you're treating every site independently, that essentially gives you some sense of, if you have, I guess that value that's saying 60% come from that parent, 40% comes from, is it already a good enough indicator? So for example, if you see, 40-60 split, then likely it's true. Can you make such an argument? Or like if you see a 95% split, then, well, who knows? Is there some,
Sorry, could you repeat one more time? I think I missed some part.
do you think that those branch probabilities are kind of indicative of, is it of the likelihood of that reticulation to be the truth? Is it like, if it's a 40-60 split, then maybe it's true because there is enough, split, and if there's a smaller split, then maybe, or do
Yeah.
you think,
Yeah, I understand. Yeah, that's actually right. And then we actually did that test. So the idea was we generated data set under a tree, and we estimated a network, letting finance know, okay, find a network with one reticulation, which is false. There is no reticulation, but we asked it to find the network with a reticulation. It gave a network topology, but this inheritance probability at the reticulation branch turns out to be 0.995 and 0.005. In this case, it might be true, there is a 0.5% of genome horizontally transferred, but it is more likely we are, overfitting. We're trying to explain the data that are better explained using trees, but we are trying to explain that using networks. Um, a better way to know this is there is a method called GHDAT, which conducts global hybridization test. So what it does is it also uses composite. It also uses site patterns and using the p values for the outputs from the quartet, the triplet based test. It tells us if the data is better explained by trees or the networks. It doesn't tell us if it's better explained by the trees how many reticulations. It doesn't tell us that, but at least it tells us it's tree versus network. So instead of conducting, like network analysis that might take a long time, probably conducting like some other analysis if it's better represented in trees or the network, and then conduct a network analysis might be a better idea. But these small inheritance parameters at the reticulation branch do informs us that if it is somewhat reliable or not.